Tonight we're going to explore something that lives in a place most human beings will never see. A place so deep and so far removed from everything we know about the surface of this planet that the creatures living there have never experienced warmth, never seen light, and have no concept of the sky above the water that separates them from us by nearly 11 kilometers of ocean. The Mariana Trench. It sits in the western Pacific Ocean, roughly 200 kilometers east of the Mariana Islands, and it is the deepest place on the surface of the Earth, a crescent-shaped scar in the ocean floor that stretches approximately 550 kilometers in length and 69 kilometers at its widest point, and reaches at its lowest known point a depth of approximately 935 meters, which is to say, if you could take Mount Everest, all 849 meters of it, and drop it upside down into the Challenger Deep, the summit would still be submerged under more than a kilometer of water, and from the surface, you would see nothing at all, nothing but the same flat, glittering expanse of Pacific Ocean that has covered this place for as long as anyone has been alive to look at it. The trench is deeper than any mountain is tall, deeper than any canyon is wide, deeper than most people ever think about when they consider the shape of the planet they live on and the life that exists at its bottom. The organisms that make their home in a place where the pressure exceeds 100 atmospheres and the temperature hovers just above freezing and the darkness is absolute and permanent, is life that challenges everything most people believe about what it takes to be alive. Most people, when they think about the deep ocean at all, think of it as empty. They think of it as a void, a black space, a place where the weight of the water squeezes out anything that might have the audacity to try to exist. And that assumption is understandable, because the conditions at the bottom of the Mariana Trench are by any human measure impossible. The pressure alone is more than 1,000 times the atmospheric pressure at sea level, equivalent to roughly 16,000 pounds pressing down on every square inch of surface, which is approximately 8 metric tons per square inch of weight, so incomprehensible that no analogy truly captures it, though it has been compared to the weight of 50 jumbo jets balanced on a single person, or the weight of 100 adult elephants standing on a surface the size of a dinner plate, or perhaps most simply, to more pressure than the human body was ever designed to encounter, more pressure than any land animal has ever experienced, more pressure than any engineer could replicate in a laboratory without highly specialized equipment that costs millions of dollars and takes years to build. By the end of tonight, you are going to understand 3 things that most people never learn. First, that the Mariana Trench is not empty. It is inhabited by organisms so perfectly adapted to the most extreme conditions on this planet that they could not survive anywhere else. Organisms that have evolved under more than 1,000 atmospheres of pressure and that would quite literally come apart at the cellular level if they were brought to the surface. The way a deep-sea fish hauled up too quickly will swell and burst as the pressure that held it together is removed. Second, that the life found at the bottom of the trench does not depend on sunlight, not even indirectly, but instead draws its energy from the chemistry of the Earth itself, from hydrogen, from methane, from sulfur, from the interior of the planet rather than the star above it, in a process so fundamentally different from photosynthesis that discovering it was like discovering a second way of being alive on the same world. And third, that this life has been there for longer than almost anything else you have ever heard of, persisting through ice ages, through mass extinctions, through the rise and fall of every civilization that has ever existed on the surface, and it will in all probability still be there long after everything above it has changed beyond recognition, because the chemistry that sustains it does not depend on climate, does not depend on seasons, does not depend on anything that happens in the atmosphere or on the continents. And it will continue for as long as the interior of the Earth remains hot enough to drive the reactions that feed it. Before we get started, if you love exploring the depths of the ocean as much as we do, take a second to like the video or subscribe. It's a simple action, but it helps this channel reach more curious minds like yours. Now, let's begin. To understand what lives at the bottom of the Mariana Trench, you first need to understand what the Mariana Trench actually is. Because the word trench can be misleading. It suggests something narrow. Something with clean walls and a flat floor like the trenches of a battlefield, or the trenches that utility workers cut into pavement to lay pipes. And the Mariana Trench is nothing like that. The Mariana Trench is a subduction zone, a place where one tectonic plate, in this case, the Pacific Plate, the largest tectonic plate on Earth, covering more than 103 million square kilometers of the planet's surface, is being forced beneath another plate, the smaller Mariana Plate. And the process that created this enormous depression in the ocean floor has been going on for more than 50 million years. The Pacific Plate carries some of the oldest oceanic crust on Earth, roughly 180 million years old, formed during the Jurassic period, when the first birds were appearing and dinosaurs still dominated every continent. And because this crust is so old, it has had a long time to cool, and because it has cooled, it has become denser and heavier than the younger, warmer plate it is colliding with, and so it sinks, bending downward into the Earth's mantle like the edge of a stiff piece of cardboard being pushed beneath a table. And the place where it bends, the place where the surface of the plate curves downward and begins its long descent into the interior of the planet, is the Mariana Trench. A tectonic plate is not a thin sheet. It is a slab of rock more than 97 kilometers thick, and bending something that thick requires forces that operate over tens of millions of years. Forces so slow and so powerful that they reshape the surface of the entire planet without any individual human being ever feeling them. The bending of the Pacific Plate at the Mariana subduction zone is not sharp or angular. It is gentle as a curve can be when it involves a piece of the Earth's crust thousands of kilometers wide. And the trench that results from this bending is not a crack in the Earth, but a fold, a place where the ocean floor curves smoothly downward into the abyss, and the abyss at its deepest point is Challenger Deep. The deepest measured point is a small slot-shaped valley at the southern end of the trench. And the most widely accepted depth measurement for this location is approximately 10,935 meters, though different surveys using different methods have produced slightly different numbers, some as deep as 10,984 meters. With a margin of uncertainty of roughly 25 meters, because measuring the depth of the ocean at these scales requires extraordinary precision. The standard method is echo sounding. A ship on the surface emits a pulse of sound downward. The sound travels through the water column, bounces off the seafloor, and returns to the ship. And the depth is calculated from the time it takes the echo to travel the round trip. But the speed of sound in seawater is not constant. It changes with temperature, with salinity, and with pressure, all of which vary along the path of the sound pulse. And a measurement error of even a fraction of a percent over a distance of nearly 11 kilometers can translate to an uncertainty of tens of meters. What is not uncertain is that the Challenger Deep is the deepest point on the surface of the Earth, deeper than any other ocean trench, deeper than any land formation is tall, and deeper than all but a handful of human beings have ever traveled by any means. One reason the Mariana Trench is so deep, deeper than the Peru-Chile Trench, deeper than the Tonga Trench, deeper than the Philippine Trench and the Kermadec Trench, and all the other subduction zones in the Pacific, is that the seafloor at this location is among the oldest in the world, and older crust is colder and denser and sinks more steeply. Another reason is that the trench lies far from any major landmass, far from the mouths of rivers that might carry sediment into the ocean and partially fill the depression the way many other deep trenches have been partly filled with sediment washed off nearby continents. The Mariana Trench sits in the middle of the open Pacific, isolated from any significant sediment source, and so it remains deep, unfilled, undiminished, as deep today as it has been for millions of years, and deepening still centimeter by centimeter as the Pacific Plate continues its slow descent into the mantle at a rate of roughly 6 to 10 centimeters per year, approximately the speed at which your fingernails grow. At the bottom of the Challenger Deep, the water column above the seafloor exerts a pressure of approximately 1,100 atmospheres, roughly 16,000 pounds per square inch. The temperature is between 1 and 4 degrees Celsius, just barely above freezing. And there is no light, because sunlight, even in the clearest tropical ocean, rarely penetrates beyond 200 meters. A depth known as the photic zone, and the bottom of the Challenger Deep is more than 50 times deeper than the last photon of visible light could ever reach. For most of the 20th century, the assumption among oceanographers and marine biologists was that nothing could live down there. The reasoning was straightforward, and it seemed at the time unassailable. The pressure would crush any biological structure, would force cell membranes into rigid, non-functional configurations, would denature the proteins that carry out the chemical reactions of life by pushing them out of their functional shapes, would make the transmission of nerve signals impossible, would prevent the expansion of lungs or the beating of a heart or any of the mechanical processes that animals depend on to survive. The cold would slow metabolic reactions to a rate so low that an organism could not generate enough energy to sustain itself. And the darkness would cut off the only source of energy that biology at the time was known to use: the sun, whose light powers photosynthesis, which powers the growth of plants, and algae, which powers everything that eats them, which powers everything that eats the things that eat them. All the way up the food chain to the top. Without sunlight, there could be no photosynthesis, and without photosynthesis, there could be no food, and without food, there could be no life. And this chain of reasoning, which seemed airtight at the time, led most scientists to conclude that the deepest ocean was biologically dead. But here's what scientists didn't realize. The logic was wrong, not because its reasoning was flawed, but because its premise was incomplete. It assumed that all life depended on sunlight. It assumed that all food chains began with photosynthesis. And it assumed that the biochemistry of surface organisms was the only kind of biochemistry that could exist. All three assumptions turned out to be incorrect, and the organisms living at the bottom of the Mariana Trench are the proof. To understand how life can exist at the pressures found at the bottom of the Challenger Deep, it helps to think about what pressure actually does to a living cell. Because pressure is not just weight, it is a force that acts on every molecule, every chemical bond, every folded protein, and every fatty acid in every membrane, and every dissolved gas in every fluid, in every cell of every organism, simultaneously and without exception. A human cell operates at a pressure of roughly 1 atmosphere, 14.7 pounds per square inch, the standard atmospheric pressure at sea level. At that pressure, the lipid bilayer that forms the membrane of every cell in your body remains in a state that biologists describe as liquid crystalline. fluid enough to allow proteins to move within it, flexible enough to change shape, permeable enough to allow nutrients and waste products to pass through specialized channels. The proteins inside the cell fold into precise three-dimensional structures, helices and sheets and loops and turns, held in place by a delicate balance of chemical forces. And these structures are what allow proteins to function as enzymes catalyzing the chemical reactions that keep the cell alive, or as channels controlling what enters and leaves the cell, or as structural components, giving the cell its shape and its mechanical integrity. Pressure disrupts all of this. As pressure increases, cell membranes stiffen, transitioning from the fluid, flexible state that they need to function into a gel-like state that is rigid and impermeable in much the same way that butter transitions from soft to hard as it cools. Proteins begin to misfold because pressure forces water molecules into the interior of the protein, disrupting the hydrophobic interactions that hold the protein's structure together. And a misfolded protein is a non-functional protein, an enzyme that cannot catalyze its reaction, a channel that cannot open a receptor, that cannot recognize its signal. Dissolved gases, particularly nitrogen and oxygen, behave differently under pressure, becoming more soluble in the blood and tissues, which is why human divers who descend too deep can experience nitrogen narcosis, a state of confusion and euphoria caused by excess nitrogen dissolving in the nervous system, or if they ascend too quickly. Decompression sickness, which occurs when dissolved gases form bubbles in the blood and tissues as the pressure drops, a condition that can be crippling or fatal. At 100 atmospheres, the pressure at the bottom of the Challenger Deep, the human body would cease to function in any meaningful sense. The lungs would collapse inward. Under the weight of the water, the chest cavity would buckle. The eardrums would have already ruptured. Thousands of meters above the gases in the bloodstream would be compressed into a state that the body was never designed to accommodate. And the nervous system, the muscles, the organs, all of them would be overwhelmed by a force so far beyond anything evolution prepared them for that the failure would be total and instantaneous. The ocean at this depth does not make exceptions. But here's what changes everything about the story of the Mariana Trench. The organisms that live at these pressures did not fall from the surface and somehow survive the transition. They did not descend from shallow water and endure the crushing increase in pressure as they sank deeper. They evolved in place over millions of years, and their biochemistry is not the same as ours. In the same way that the biochemistry of an organism living in boiling hot springs is not the same as the biochemistry of an organism living in a temperate forest. Evolution shaped them for their environment, not for ours. And the result is a biology that is, in many fundamental ways, incompatible with life on the surface. Their cell membranes contain different lipids, specifically higher proportions of unsaturated fatty acids, which have kinks in their hydrocarbon chains that prevent them from packing together tightly, keeping the membrane fluid and functional at pressures that would solidify a surface cell membrane the way cold solidifies cooking oil in a refrigerator. Their proteins fold differently, stabilized by chemical bonds and structural arrangements that are strengthened rather than disrupted by extreme pressure, so that these proteins actually function better at the bottom of the Mariana Trench than they would at the surface. The way a stone arch becomes more stable, not less, when weight is placed on top of it, because the geometry of the structure converts the downward force into lateral compression that holds the stones more firmly in place. And many of the deepest living organisms produce specialized molecules called piezolites, chemical compounds whose specific function is to protect proteins from the damaging effects of pressure. The most important of these piezolites is trimethylamine N-oxide, or TMAO, a small molecule that accumulates in the cells of deep-sea organisms in concentrations that increase with depth, forming a linear relationship so consistent and so predictable that scientists can estimate the maximum depth at which a given species can survive, simply by measuring its cellular TMAO concentration. The way an engineer can predict the performance ceiling of an engine by measuring the properties of its fuel. TMAO works by stabilizing the three-dimensional structure of proteins. It preferentially interacts with water molecules in a way that makes it thermodynamically unfavorable for water to penetrate the interior of a folded protein, effectively shielding the protein from the unfolding effect of pressure. The deeper the organism lives, the more TMAO it needs. And for fish, vertebrates with complex nervous systems and specialized organs and protein structures that are inherently more sensitive to pressure perturbation than those of simpler organisms. This creates an absolute biochemical ceiling. At approximately 8,200 to 8,400 meters, the concentration of TMAO required to stabilize vertebrate proteins becomes so high that it begins to interfere with other cellular processes, creating a physiological contradiction that no amount of further adaptation can resolve. Below that depth, there are no fish, and there will never be fish, because the chemistry of the vertebrate cell simply cannot be made to work at those pressures. But below that depth there is life, abundant, diverse, and in many cases thriving, because the organisms that live below the fish line are not vertebrates, and their biochemistry plays by different rules. And still, we have not reached the bottom. The first creatures you would encounter near the bottom of the Mariana Trench, if you descended in a submersible with lights powerful enough to illuminate the seafloor, would almost certainly be amphipods, small, translucent shrimp-like crustaceans that drift and swim and crawl through the darkness in numbers that would surprise you, because the Mariana Trench, for all its apparent inhospitality, supports dense populations of these animals at depths that would destroy almost any other multicellular organism on the planet. Amphipods are crustaceans related to the sandhoppers you might find on a beach or the tiny, darting creatures that swim in the shallows of a freshwater stream, but the amphipods of the Hadal zone, the ocean zone below 6,000 meters, named after Hades, the Greek god of the underworld, are as different from their surface relatives as a polar bear is from a house cat. They share a common ancestor, but tens of millions of years of evolution in the deepest, darkest, most pressurized environment on the planet have reshaped them into something the surface relatives would not recognize. Most hadal amphipods are small, some no larger than a grain of rice, others a centimeter or two in length. But some species, the so-called supergiant amphipods, can grow to lengths of more than 5 centimeters, which may not sound large until you consider that a 5-centimeter amphipod at 10,000 meters depth is proportionally one of the most successful large-bodied organisms in the most extreme environment on the planet. During the Hades expedition to the Mariana Trench in 2014, led by researchers from the University of Hawaii, aboard the research vessel Falcor, scientists documented the highest density of supergiant amphipods ever seen. Swarming around baited camera landers lowered to the trench, floor their pale, almost ghostly bodies moving with a deliberate, purposeful energy that seemed out of place in a world so hostile. These amphipods are scavengers. They feed on organic material that falls from the water column above. The slow, constant rain of dead plankton fecal pellets shed mucus, the cast-off skins of larvae, and occasionally the carcass of a larger animal that has died in the productive waters near the surface. and sunk through the water column, falling for days or weeks through progressively darker, colder, higher pressure water until it finally comes to rest on the sediment at the bottom. This rain of organic material is what oceanographers call marine snow, and it is the primary food source for most hadal organisms. The slender thread that connects the deepest life on Earth to the sunlit surface, thousands of meters above. But by the time marine snow reaches the bottom of the Mariana Trench, it has been falling for a very long time. A particle leaving the surface must travel through more than 10,000 meters of water, and during that descent it is consumed and reconsumed by organisms at every depth level. Bacteria break it down. Zooplankton feed on it, gelatinous organisms trap it in mucus nets, and each successive consumer takes a portion of the energy it contains, so that by the time the particle finally settles on the trench floor, it is depleted. Exhausted, a shadow of the nutritious morsel it was when it began its descent. Some estimates suggest that only 1 to 3% of the organic material produced at the surface ever reaches the abyssal seafloor at depths of 4,000 to 5,000 meters. And the fraction that reaches the hadal zone below 6,000 meters is smaller still. And yet, the amphipods thrive. They detect food particles on the sediment using chemical receptors that are exquisitely sensitive, capable of sensing organic molecules in the water from considerable distances. And they converge on food sources with a speed and coordination that seems improbable for organisms living in an environment that imposes such extreme metabolic costs. When researchers lower baited camera landers to the bottom of the Mariana Trench, instruments that carry a piece of fish or mackerel as bait, attached to a camera and light system that records whatever approaches, the amphipods arrive within minutes, sometimes within seconds, swarming around the bait in dense aggregations that can number in the hundreds or thousands, their bodies flickering in the camera lights as they tear at the food with their mandibles, consuming it with an efficiency that leaves almost nothing behind. The adaptations that allow hadal amphipods to survive at extreme depth are molecular. Their cells contain high concentrations of TMAO, the same piezolyte that protects fish proteins at depth, but they also contain other protective molecules that are less common in vertebrates. Glycerophosphorylcholine, which is found at high concentrations in mammalian kidneys, where it protects proteins from the osmotic stress of waste products, and silo-inositol, a molecule that is currently being studied for its potential to restore misfolded proteins in the brains of patients with Alzheimer's disease. The fact that these same molecular protectors are found in human kidneys and in the cells of amphipods at the bottom of the Mariana Trench is one of those quiet connections that biology occasionally reveals, a reminder that the chemistry of life, no matter how extreme the environment, draws from the same molecular toolkit, the same set of solutions to the same set of problems, separated by billions of years of evolution, but still recognizably related. Researchers at the Schmidt Ocean Institute, during an expedition to the Mariana Trench aboard the research vessel Falkor in 2014, discovered that one species of hadal amphipod possesses an enzyme capable of digesting wood, cellulose, the tough structural material that gives plant cells their rigidity, an enzyme that functions better under high pressure than it does at atmospheric pressure. This is not an enzyme that merely tolerates pressure. It is an enzyme that is optimized for it, that performs its function more efficiently at more than 1,000 atmospheres than it does at one, as though the organism had been designed. Though of course it was not designed, but evolved slowly over millions of years to work best at the very conditions that would kill almost any surface organism. For these amphipods, the crushing pressure of 1,100 atmospheres is not an obstacle. It is not a hardship. It is the condition under which their biology works best, the condition for which their proteins are folded, their membranes are tuned, their enzymes are calibrated. The warm, low-pressure world where we live, the one-atmosphere world of sunlight and air and mild temperatures, would be as lethal to them as the bottom of the Challenger Deep would be to us, because their cellular machinery would simply come apart without the pressure that holds it together, the way a submarine collapses inward if it descends too deep, but would also, in a different way, fail if it were suddenly placed in a vacuum. For these amphipods, pressure is not the enemy. Pressure is the medium in which they exist, and without it, they would cease to exist entirely. For them, this environment is not extreme. For them, this is home, and the world above, the warm, bright, low-pressure world where we live, would be as hostile to them as the deep ocean is to us. This is the perspective inversion that the Mariana Trench demands of anyone who tries to understand it. That extreme is relative, that habitable is a matter of adaptation, and that the organisms at the bottom of the trench are not struggling to survive in a hostile environment. They are thriving in the only environment that is suitable for them. Keep going. Below the amphipods, in the sediment of the trench floor itself, live organisms that challenge the very definition of what it means to be a single creature. These are the xenophyrophores, and they are, by most measures, the largest single-celled organisms on Earth. A xenophyrophore is a single cell. One cell. Not a colony of cells working together. Not a tissue composed of specialized cell types, not an organ or an organism, in the way that we typically use those words. A single unified cell with no internal organs, no nervous system, no muscles, no brain. No specialized tissues of any kind, just a mass of cytoplasm, containing many nuclei flowing through a network of organic tubes, called granulae, all enclosed within an external shell, called a test, that the organism builds particle by particle, grain by grain, from materials it gathers from the sediment around it, and despite being a single cell. Some specimens can reach up to 20 centimeters in diameter, roughly the size of a mango or a human fist, making them tens of thousands of times larger than a typical animal cell, which is generally around 10 to 30 micrometers, across a size so small that it is invisible to the naked eye and requires a microscope to observe. The largest known species, Syringamina fragilissima, can reach up to 24 centimeters across, making it among the largest single cells ever documented in the history of life on this planet. In 2011, a research expedition organized by the Scripps Institution of Oceanography at the University of California, San Diego, led by marine microbiologist Doug Bartlett, used unmanned high-definition cameras called drop cams, developed in collaboration with National Geographic engineers, to explore the depths of the Mariana Trench. The expedition recorded xenophyrophores at depths of up to 641 meters in the Serena Deep, a basin within the Mariana Trench, setting the deepest known record for these organisms at the time, and extending the known depth range of the group by nearly 2 miles beyond the previous record of approximately 7,500 meters, which had been set in the New Hebrides Trench. Lisa Levin, a Scripps professor and deep-sea biologist, described the xenophyrophores as highly adapted to extreme conditions, but at the same time very fragile and poorly studied, which captures the fundamental paradox of these organisms. They are among the most resilient life forms on the planet in terms of the environments they can inhabit. Yet they are so physically delicate that specimens invariably arrive at the surface as mangled, useless fragments, making laboratory study nearly impossible. The name xenophyrophore comes from the Greek and means bearer of foreign bodies, which is a precise description of how these organisms build their tests. A xenophyrophore does not grow a shell the way a snail or a clam grows a shell, secreting calcium carbonate from its own tissues to form a hard, uniform structure. Instead, it gathers material from its surroundings. Grains of sand, fragments of the shells of other foraminiferans, sponge spicules, mineral particles, bits of volcanic glass. The skeletal remains of radiolarians, and cements them together with organic secretions to form structures that can take an extraordinary variety of shapes. Fans, spheres, flat discs, branching networks of tubes. Ridged masses that resemble the surface of a brain, and lumpy irregular mounds that look like nothing in particular. One species remarkably builds its test exclusively from the glass spicules of deep-sea sponges, constructing a glittering transparent house from the skeletons of its neighbors, a structure that is in its own quiet way as architecturally distinctive as anything built by creatures millions of times more complex. Xenophyrophores belong to the foraminifera, a group of amoeba-like protists with a fossil record that extends back more than 500 million years deep into the Cambrian period, when the first complex animal life was just appearing on Earth. Approximately 75 species of xenophyrophores have been described across 15 genera, and they have been found in all 4 major ocean basins: the Atlantic, the Pacific, the Indian, and the Southern, at depths ranging from roughly 500 meters to more than 600 meters. They are absent only from the Arctic, and they are found in almost every deep-sea environment that offers the right combination of sediment, organic carbon supply, and stable conditions. On abyssal plains, on the slopes of seamounts, in submarine canyons, on continental margins, and in the Hadal trenches. In some regions, they are the dominant visible organisms on the seafloor, more abundant and more conspicuous than any animal in the same habitat. What makes xenophyrophores ecologically important is not just their size or their abundance, but their role as ecosystem engineers. The deep sea floor, particularly in the abyssal and hadal zones, is one of the most featureless landscapes on Earth. Mile after mile of flat, soft sediment with almost no topographic relief, no rocks, no ridges, no structures of any kind to break the monotony. In this environment, the tests of xenophyrophores provide some of the only hard surfaces available, and other organisms, brittle stars, isopods, polychaete worms, nematodes, tiny crustaceans, cluster around them and on them, using them as habitat in the same way that organisms in shallow water use coral or rocky outcrops. Studies conducted by Levin and her colleagues have shown that areas of the deep-sea floor where xenophyrophores are present have measurably higher biodiversity, more species, more individuals, more ecological complexity than similar areas without them, which means that these single-celled organisms, each one a single cell, no more complex in its basic biology than the amoebas that live in pond water, are functioning as the keystone organisms of their ecosystems, providing habitat and structure in a world that would otherwise offer none. They feed on what they can gather from the sediment, detritus microbes, dissolved organic matter, using their pseudopodia, the flowing extensions of cytoplasm that amoeba-like organisms use to move and to engulf food particles. And they are thought to cultivate bacterial colonies within their own waste products, growing microbes on their fecal pellets and then consuming them, a form of microscopic farming that has only recently been confirmed through laboratory analysis. They also concentrate heavy metals, lead, uranium, mercury, in their tests, accumulating these toxic elements to levels that would kill most organisms apparently without harm, a capacity whose ecological function is still not understood. They were there before the dinosaurs appeared, roughly 230 million years ago. They were there before the first mammals, before the first birds, before the first flowering plants. They were there when the supercontinent Pangaea was breaking apart, when the Atlantic Ocean was just a narrow crack between Africa and South America, when the Indian subcontinent was still a separate island drifting northward toward its eventual collision with Asia. They were there when the first primates appeared roughly 60 million years ago, and when the first hominids stood upright roughly 4 to 6 million years ago, and when the first modern humans looked out at the ocean and wondered what lay beneath it roughly 300,000 years ago. And they are still there now, on the floor of the Mariana Trench, building their tests from the sediment, grain by grain, cell by single cell, in a process that has continued uninterrupted for longer than any land animal lineage has existed. But we are still not deep enough in the story. And if the xenophyrophores are the architects of the trench floor, there is another organism that inhabits these depths, in a form so alien, so removed from anything the surface world would recognize, that it is worth pausing to consider. The foraminifera of the Hadal zone. The smaller relatives of the xenophyrophores are among the most abundant organisms in the deep-sea sediment, and their shells, their tests, have been accumulating on the ocean floor for hundreds of millions of years, forming layers of calcareous ooze that can be hundreds of meters thick, a geological record of deep-sea life, written in the language of microscopic shells, each one a few hundred micrometers across. Each one the remains of a single-celled organism that lived and died in the deep ocean at some point in the last several hundred million years. The sediment at the bottom of the Mariana Trench is different from the sediment on most of the ocean floor, because the extreme depth and the high pressure affect the chemistry of calcium carbonate, the material from which most foraminiferan shells are made. Below a depth, known as the calcite compensation depth, typically around 4,000 to 5,000 meters in the Pacific, the water becomes so corrosive to calcium carbonate that shells dissolve faster than they accumulate, and the sediment transitions from calcareous ooze to a fine, clay-like material called red clay or pelagic clay, composed primarily of the insoluble remains of wind-blown dust volcanic ash and the siliceous skeletons of radiolarians and diatoms. At the bottom of the Challenger Deep, the sediment is predominantly this fine clay mixed with the siliceous remains of organisms whose glass-like shells are resistant to dissolution, and it is in this material, this ancient, slowly accumulating blanket of the finest possible particles, that the xenophyrophores build their tests, and the amphipods search for food, and the microbes carry out their geological scale metabolism. The environment of the trench floor is in many ways defined by what is absent rather than by what is present. There is no light. There is almost no current. The water at the bottom of the Challenger Deep moves so slowly that it takes centuries for a given mass of water to be replaced by the sluggish, density-driven circulation of the deep ocean. There is almost no food relative to the productive surface waters above. The marine snow that reaches the bottom has been depleted by organisms at every level of the water column. And what arrives at the trench floor is nutritionally impoverished, the equivalent of crumbs swept from a table where the meal has already been eaten by 100 diners before the food reaches the floor. And there is almost no structure, no rocks, no ridges, no topographic features that an organism could use as shelter or as a substrate for attachment. Just flat, soft, featureless mud stretching in every direction. Kilometer after kilometer, as uniform and as empty as any landscape on Earth. And yet, In this apparent emptiness, life persists. Not in the exuberant, competitive, visually dramatic way that life persists on a coral reef, where every square centimeter is contested and every available surface is colonized within days, but in a slower, quieter, more patient way. A way that is defined by scarcity rather than by abundance. By persistence rather than by competition, by the ability to extract the last possible calorie from the last possible particle of food, and to survive on that calorie for as long as it takes for the next one to arrive. The organisms of the Mariana Trench do not live fast. They do not die young. They live slowly, they endure, and they wait. And the waiting is not passive, it is an active, metabolically expensive process that requires energy and vigilance and the continuous maintenance of cellular structures under conditions that would destroy any surface organism within seconds. Survival at the bottom of the Mariana Trench is not the absence of effort. It is effort of a different kind, the effort of persistence, of continuity, of holding together in a world that is trying at every moment to compress you into nothing. And still, we have not reached the strangest inhabitants of this place. Because there is a fish in the Mariana Trench, a small, pale, seemingly impossible fish, that represents the absolute boundary of vertebrate life on Earth. This is the snailfish, and its story is one of the most remarkable in all of deep-sea biology. Snailfish belong to the family Liparidae, a family of more than 400 known species that live in oceans around the world, from shallow tide pools to the deepest trenches, from the warm waters of the tropics to the freezing seas around Antarctica. They are not particularly impressive to look at. Small, soft-bodied, without scales, often translucent or pale, with round heads and tapered bodies that give them a vaguely tadpole-like appearance. And they are not well known outside of the scientific community, because most people have never heard of them, and probably never will. But in the Hadal Zone, snailfish are the apex predators, the top of the food chain, the deepest diving vertebrates on the planet. And their adaptations to extreme depth are among the most sophisticated that evolution has ever produced in a vertebrate organism. In the Mariana Trench, the species Pseudoliparis swirae, formally described in 2018, based on specimens collected during the Hades expedition, has been observed at depths of more than 8,000 meters, and genetic analysis published in the journal Nature, Ecology and Evolution has shown that this species diverged from its closest known relative, a shallow water snailfish living near the sea surface, approximately 20 million years ago. 20 million years. That is roughly 4 times longer than the genus Homo has existed, 4 times longer than any recognizable ancestor of modern humans has walked upright on the surface of the Earth. The Mariana snailfish has been evolving for the deep since long before humanity was even a possibility. The fish itself is small, typically no more than about 20 centimeters long, and its body has been reshaped by 20 million years of natural selection for life at extreme depth. It has no scales. Scales are rigid structures, and rigidity is a liability at extreme pressure, where flexibility allows the body to compress slightly and absorb the force rather than cracking. Its skin is transparent, almost ghostly, revealing the outlines of its internal organs, the liver, the stomach, the gonads, through the thin, unpigmented tissue. There is no reason to produce pigment in a world without light, and so the snailfish has stopped producing it, conserving the metabolic energy that pigment synthesis would require and redirecting it to more essential functions. Its eyes are tiny, reduced to small dots on the sides of its head that may retain some sensitivity to the faint bioluminescent flickers produced by other organisms in the deep, but that are largely vestigial. Evolutionary remnants of a time when the snailfish's ancestors lived in water where light still existed. Its skull is not fully ossified, meaning that the bones of its head never fully harden, remaining soft and cartilaginous in a way that is unusual for an adult fish. A typical surface fish has a fully hardened skull by the time it reaches maturity, but the Mariana snailfish retains a partially open, unfused cranium throughout its life. And genetic analysis has revealed that this is not a developmental anomaly, but the result of a specific genetic change, a frameshift mutation in the bone glar protein gene, which normally directs the calcification of cartilage into bone, causing an early termination of the calcification process and leaving the skull permanently soft. This softness may serve a specific survival function, because a flexible skull can compress slightly under extreme pressure, rather than cracking, absorbing the force the way a rubber ball survives being squeezed, while a glass ball shatters under the same force. Its stomach is inflated disproportionately large for its body size, which may function as a storage organ for food in an environment where meals are unpredictable and may be separated by long intervals of fasting. But the deepest fish ever filmed was not in the Mariana Trench. In September 2022, during a 2-month expedition led by Professor Alan Jameson of the University of Western Australia's Minderoo Deep Sea Research Centre, in collaboration with the Tokyo University of Marine Science and Technology, a team filmed an unknown species of Pseudoliparis snailfish at a depth of 8,336 meters, approximately 27,350 feet, more than 5 miles below the surface, in the Izu Ogasawara Trench off Japan. This depth is approximately 158 meters deeper than the previous deepest fish recording, which had been made by the same team in 2017 at 8,178 meters in the Mariana Trench. Days after the filming, the team physically caught 2 specimens of Pseudoliparis belliaevi from traps set at 8,022 meters in the nearby Japan Trench, making them the first fish ever collected from below 8,000 meters, a watershed moment in deep-sea biology, the biological equivalent of collecting a rock from the summit of an unclimbed mountain. The reason the deepest fish was found in the Japanese trenches rather than in the deeper Mariana Trench has to do with temperature. The Japanese trenches are warmer than the Mariana, fed by warmer currents that circulate through the Western Pacific, and temperature and pressure have similar perturbing effects on cellular proteins. Both cause proteins to unfold, to lose the 3-dimensional shapes that make them functional. In warmer water, more thermal energy is already destabilizing the proteins, which means that less TMAO is needed to counteract the combined effects of temperature and pressure, and the organism can therefore tolerate slightly greater depth before its total protein stabilizing budget is exhausted. As Jameson explained, temperature is not just a secondary factor. It is a critical variable that determines, alongside pressure, the absolute depth limit for vertebrate life. Scientists now believe that the maximum depth at which any fish can survive is somewhere between 8,200 and 8,400 meters. Below that, the concentration of TMAO required to stabilize vertebrate proteins exceeds what the cells can accommodate without disrupting other essential processes. The TMAO itself begins to interfere with the very chemistry it is supposed to protect, creating a biochemical paradox that cannot be resolved by further adaptation. Below 8,400 meters, the hadal zone belongs entirely to the invertebrates, to the amphipods, the sea cucumbers, the xenophyophores, the polychaete worms and the microbes, and the fish simply cannot follow. There is a line invisible and absolute drawn, not by geography, but by biochemistry below, which no vertebrate has ever been found, and, in all likelihood, no vertebrate will ever be found, because the physics of protein chemistry at those pressures does not permit it. As Jameson has observed, each deep-sea trench appears to have its own species of snailfish. The Mariana Trench has Pseudoliparis swirae. The Kermadec Trench has a different species. The Japan Trench has Pseudoliparis bellaevi. And so on. And once a snailfish lineage has evolved to cope with the extreme conditions of its home trench, it cannot decompress to cross the shallower water between trenches and colonize another. The trench is a one-way adaptation. The fish that descend into it over evolutionary time become specialized for that trench, and that trench alone, and their descendants stay. Locked in by walls of pressure, as impassable as any mountain range, evolving in isolation, accumulating genetic differences, becoming endemic, found nowhere else in the world. In the same way that the species of the Galapagos Islands became unique to their particular island, not because they were geographically distant from the mainland, but because the ecological barrier between them and everything else was for all practical purposes insurmountable. The snailfish feeds on amphipods, the same small crustaceans that swarm the sediment below, the same organisms that gather around baited landers in their hundreds and thousands. In the Mariana Trench, at depths between 7,000 and 8,000 meters, snailfish have been observed in surprising numbers gliding slowly over the pale mud, their translucent bodies almost invisible against the light-colored sediment. Their wing-like fins barely moving as they drift through water that has not seen sunlight in 1,000 years or more. They hunt not by sight, but by vibration, by chemical sensitivity, by the lateral line system, a row of sensory organs along the body that detects pressure waves and movements in the water, a sense that functions in total darkness as effectively as vision functions in light. For these fish, the darkness is not a limitation. It is the only world they know, a world their ancestors would not recognize, because those ancestors could see and lived in light. But over 20 million years, as the lineage moved deeper, as the light disappeared, as the eyes became smaller and less functional, the snailfish traded one sense for another. And the world it lives in now, a world of pressure waves and chemical gradients and faint vibrations in the water, is for the snailfish as rich and as navigable as the sunlit world is for us. And still, we have not reached the bottom. There is a dimension of the Mariana Trench that changes everything about how we understand life on this planet. And it begins with a simple question. Where does the energy come from? On the surface of the Earth and in the upper layers of the ocean, the answer is straightforward. The energy comes from the sun. Sunlight strikes the surface of the ocean, or the leaves of a plant, and photosynthetic organisms, algae, phytoplankton, cyanobacteria, land plants, capture that energy and use it to convert carbon dioxide and water into organic carbon, the molecular building material from which all living tissue is constructed. Every salad you have ever eaten, every grain of rice, every piece of bread, every glass of milk, every steak, every apple, every fish on every plate, in every restaurant in the world, all of it traces its energy back to the sun, through a chain of organisms that begins with a photosynthetic producer and ends with whatever ate last. Even the marine snow that feeds the amphipods in the Mariana Trench is in most cases derived from photosynthesis. It consists of dead phytoplankton fecal pellets, from organisms that ate phytoplankton shed mucus, from organisms that filtered phytoplankton from the water, and the occasional carcass of an animal whose energy, if you traced it back far enough through the food web, originated in the sunlit surface waters of the ocean. But at the very bottom of the Mariana Trench, there is another source of energy, one that has nothing to do with the sun, nothing to do with photosynthesis, nothing to do with anything that happens on the surface of the earth, or in the atmosphere, or in the sky above it. This is chemosynthesis, the process by which organisms derive energy from chemical reactions rather than from light, and its discovery was one of the most important findings in the history of biology, because it proved that life does not require a star. The story of chemosynthesis begins in 1977 at the bottom of the Pacific Ocean, roughly 500 meters below the surface, at a location called the Galapagos Rift, a divergent plate boundary in the eastern Pacific, where the Nazca Plate and the Cocos Plate are spreading apart. A new oceanic crust is being formed. A team of geologists aboard the research submersible Alvin, a small, 3-person deep-sea vehicle operated by the Woods Hole Oceanographic Institution, in service since 1964, descended to the rift to study the geology of the spreading center, expecting to find bare rock, fresh lava, and perhaps some mineral deposits around the fissures where hot water was thought to emerge from the crust. What they found instead was life. Dense, abundant, wildly improbable life, thriving in total darkness around fissures in the seafloor, where superheated mineral-laden water was erupting from the Earth's crust in plumes of black and white fluid that looked like underwater smoke. The water from these vents was extraordinarily hot. Temperatures exceeding 400 degrees Celsius were measured at some sites. Though the water did not boil, because the immense pressure at that depth keeps it in a liquid state, and it was loaded with dissolved minerals and gases, particularly hydrogen sulfide methane, and various metal compounds that gave the vent fluid its characteristic dark color. Around these vents, the scientists found ecosystems unlike anything ever described in the scientific literature. Giant tube worms, later named Riftia pachyptila, stood in dense clusters around the vents, some of them more than 2 meters long, their red feathery respiratory plumes extending from white tubes anchored to the rock. Clams with shells as large as dinner plates were wedged into cracks in the lava. Mussels carpeted the rocks. Shrimp swarmed in the warm water near the vent openings. White crabs picked their way across the mineral-encrusted surfaces. And bacteria, enormous mats of white filamentous bacteria, blanketed the rocks near the vents, forming thick, ghostly carpets of microbial growth that covered every available surface. None of these organisms were eating anything from above. None of them were connected to the surface food web. None of them depended, even indirectly, on sunlight. The base of their food chain was chemosynthesis, specifically the oxidation of hydrogen sulfide by bacteria that use the energy released by this reaction to fix carbon dioxide into organic carbon, in a process that is chemically analogous to photosynthesis, but that uses chemical energy instead of light energy. These chemosynthetic bacteria were the primary producers of the vent ecosystem, the equivalent of plants and algae in the surface world and everything else in the ecosystem. The tube worms, the clams, the mussels, the shrimp, the crabs depended on them, either by eating them directly or by harboring them as symbionts inside their own bodies. The giant tube worms were the most dramatic example. When scientists dissected them, they found that Riftia pachyptila had no mouth, no stomach, no digestive tract of any kind. The worm could not eat. Instead, it harbored billions of chemosynthetic bacteria inside a specialized organ called a trophosome, which filled most of the worm's interior. The worm's red plumes, colored red by hemoglobin. the same oxygen-carrying protein found in human blood, absorbed hydrogen sulfide oxygen and carbon dioxide from the vent water and transported them via the bloodstream to the trophosome, where the bacteria performed chemosynthesis and produced the organic carbon that fed the worm. The worm was not eating the bacteria. The bacteria were not parasitizing the worm. They were living in a symbiosis, so complete, so deeply integrated at the molecular level that neither organism could survive without the other. A partnership that had been evolving and refining itself for hundreds of millions of years. The discovery of hydrothermal vent communities in 1977 changed biology. It proved that life could exist without sunlight. It proved that ecosystems could function independently of photosynthesis. It proved that the energy required to sustain living organisms could come from the interior of the Earth rather than from the star at the center of the solar system, and it raised the possibility, still debated, but increasingly supported by evidence, that life on Earth may have originated not in the sunlit shallows of a warm ocean, as had long been assumed, but at hydrothermal vents on the deep sea floor, where the chemistry of the Earth's interior provided both the energy and the raw materials for the first self-replicating molecules. But here's what most people never realized. For decades after the discovery of hydrothermal vents, scientists assumed that chemosynthetic communities were confined to specific geological settings: active vent sites on mid-ocean ridges, cold seeps on continental margins, and a few other isolated locations where the right combination of chemicals and geological activity existed to support this alternative form of life. The Hadal Trenches, including the Mariana Trench, were thought to be too deep, too remote, too far from the tectonic boundaries where vents typically form to support chemosynthetic ecosystems. This assumption was wrong. The Mariana Trench is itself a zone of intense geological activity. The subduction of the Pacific Plate beneath the Mariana Plate generates heat, deforms rock, and releases enormous volumes of water and dissolved gases from the sinking crust. As the ancient oceanic plate descends into the mantle, the minerals in its rock, serpentine, olivine, pyroxene, react with the water trapped in their crystal structures in a process called serpentinization, releasing hydrogen, methane, and other reduced chemical compounds that rise through fractures in the overriding plate and seep out through the seafloor, creating conditions that are in many ways directly analogous to those found at hydrothermal vents and cold seeps at shallower depths. The trench is not geologically quiet. It is one of the most geologically active places on the planet, and the chemistry of that activity, the methane, the hydrogen, the sulfur compounds, sustains life. In 2025, a team of researchers using the Chinese manned submersible Fenduji, which had first reached the bottom of the Challenger Deep in November 2020, descending to a depth of 909 meters, published findings in the journal Nature that fundamentally changed the understanding of life in the deepest parts of the ocean. Diving to multiple sites along the floors of several Hadal trenches, including the Mariana Trench and the Kuril-Kamchatka Trench, the team discovered extensive communities of chemosynthesis-based organisms at depths ranging from 5,800 to 9,533 meters, the deepest and most extensive chemosynthetic communities ever observed anywhere on Earth. These were not isolated patches of bacterial mats or scattered individual organisms at the edge of survival. These were dense, thriving aggregations of tube worms and bivalves, hundreds and thousands of individuals living in and on the sediment, clustering around areas where methane, produced by microorganisms deep in the subsea floor, was seeping up through the mud. The tube worms belonged to the family Siboglinidae, the same family as the giant vent worms of the Galapagos Rift. And they were found at densities of more than 500-800 individuals per square meter, concentrations rivaling the most productive vent communities known anywhere on the planet. The bivalves, vesicomyid clams, were present at densities of nearly 300 per square meter, their shells partially buried in the sediment. Their tissues, harboring chemosynthetic bacteria that metabolized the methane and hydrogen sulfide seeping from below. The researchers found compelling evidence that the methane was being produced not by geological processes, but by microbial activity deep in the subsea floor sediments, with the tube worms clustering around microbial mats that resembled patches of white snow on the dark mud, mats of bacteria and archaea that were themselves the chemical factories feeding the entire community. The researchers noted that similar geological conditions, subduction, serpentinization, fluid release, microbial methane generation, exist at dozens of other hadal trenches around the world, and they suggested that such chemosynthesis-based communities might be far more widespread than anyone had previously imagined, waiting to be found in places that no human instrument has yet been able to reach. This is the center of tonight's subject, the point where the facts become something more than facts. The organisms at the bottom of the Mariana Trench have never seen the sun. They do not know the sun exists. The sun, for them, is irrelevant, a distant object that has no bearing on their survival, no connection to their food web, no influence on their biology. They are sustained by the earth itself, by the chemistry of the rocks beneath them, by the methane produced by microbes that eat minerals in the sediment, by the hydrogen released when water reacts with olivine in the subducting plate, by processes that began when this planet was young and molten, and that will continue for as long as the interior of the Earth remains hot enough to drive them, which will be, by current estimates, for at least another billion years. And they have been living this way for a very long time. The fossil record of chemosynthetic communities extends back at least 400 million years, and molecular evidence suggests that chemosynthesis itself may be one of the oldest forms of energy production on Earth, possibly predating photosynthesis by hundreds of millions of years. If this is true, and the evidence grows stronger with each decade of research, then the organisms at the bottom of the Mariana Trench are not the outliers. They are not the curiosities at the margin of biology. They are practicing the original way of being alive, the ancestral metabolism, the first solution that life found to the problem of obtaining energy from a world that was 4 billion years ago, dark and hot and covered in water and utterly devoid of oxygen. Everything that has happened since, all the forests, all the grasslands, all the animals, all the civilizations, is the newcomer, the experiment that came after. Keep going. There is a geological dimension to the life at the bottom of the Mariana Trench that deserves its own consideration, because the trench is not just a habitat, it is a geological machine, and the organisms that live in it are participants, however unwitting, in one of the largest and most powerful processes operating on the surface of the Earth. The Pacific Plate is subducting beneath the Mariana Plate at a rate of roughly 6 to 10 centimeters per year, which means that the seafloor at the bottom of the Mariana Trench is moving, slowly, imperceptibly, but continuously toward the interior of the planet. The sediment on which the amphipods are feeding tonight will be carried over millions of years into the mantle, where it will be heated, compressed, melted, and eventually return to the surface as new volcanic material at a mid-ocean ridge somewhere else in the world, millions of years from now. The organisms at the bottom of the trench are living on a surface that is moving beneath them like passengers on a conveyor belt, so slow that no individual organism could detect its motion but over geological time. The motion is relentless and irreversible. The trench is not a fixed place, it is a process. The subduction of the Pacific Plate has been occurring at this location for more than 50 million years, and during that time, an enormous volume of oceanic crust, thousands of kilometers of rock, roughly 97 kilometers thick, has been pushed down into the mantle, carrying with it the sediment, the water, the chemicals, and, in all probability, the organisms that were living on it at the time. Seismologists studying the earthquakes generated by this subduction zone have traced the path of the descending plate to depths of more than 700 kilometers below the surface, where it enters the lower mantle and is slowly absorbed back into the hot convecting rock from which it originally formed, completing a cycle of creation and destruction that has been operating since the earliest days of plate tectonics, roughly 3 to 4 billion years ago. The material that forms the Pacific Plate at the Mariana Trench is approximately 180 million years old. It was formed during the Jurassic period, when Stegosaurus and Allosaurus roamed the continents and the supercontinent Pangaea was beginning to break apart. Since its formation at a mid-ocean ridge in the central Pacific, this crust has traveled thousands of kilometers across the ocean floor, accumulating a thin blanket of sediment as it went. The remains of trillions of organisms that lived and died in the water above, settling slowly onto the rock surface at a rate of roughly 1 centimeter per 1,000 years, century after century, millennium after millennium, for 180 million years. And now at the Mariana Trench, this ancient crust is reaching the end of its passage across the ocean floor, bending downward, descending into the interior of the planet, carrying its blanket of sediment and its burden of water and its community of deep-sea organisms with it. This is a perspective that few people ever encounter, and it is worth sitting with for a moment. The rock at the bottom of the Mariana Trench has been traveling across the surface of the Earth for 180 million years, slowly and continuously, and during that time, the entire world has changed around it. Continents have separated and collided, oceans have opened, and closed species have evolved and gone extinct by the millions. The climate has swung from hothouse to icehouse and back again, and through all of it, this rock has been moving, accumulating sediment, cooling, aging, and now finally arriving at the subduction zone, where it will be consumed. The organisms living on it, the amphipods, the xenophyophores, the microbes in the sediment, are living on a surface that is older than any continent, older than any mountain range, older than any ecosystem on land, and they are riding it unknowingly into the interior of the Earth. But the subduction does not just destroy, it also creates. As the Pacific Plate descends, the water trapped in its minerals, water that has been locked into the crystal structure of the rock for tens of millions of years, is released by the increasing heat and pressure, and it rises through fractures in the overriding plate, triggering volcanic activity that builds the Mariana Islands, the chain of volcanic peaks that rises from the seafloor to the west of the trench. The Mariana Islands exist because the trench exists. The volcanoes that formed them were powered by the water released from the subducting plate, and the magma that erupted from those volcanoes was generated by the melting of the mantle as that water passed through it. The trench and the islands are two expressions of the same process, one a place of destruction, the other a place of creation, and both are powered by the slow, relentless descent of 180-million-year-old rock into the interior of the planet. And it is this same process, the release of water and gases from the subducting plate, that feeds the chemosynthetic communities at the bottom of the trench. The serpentinization reactions that produce hydrogen and methane in the overriding plate are driven by the fluids released from the descending plate, and the organisms that metabolize these chemicals are, in a very real sense, fed by the same geological process that builds the islands and generates the earthquakes and drives the entire tectonic system. The life at the bottom of the Mariana Trench is not separate from the geology. It is part of the geology. It is an expression of the same planetary scale processes that move continents, create mountains, and shape the surface of the Earth, and it will continue for as long as those processes continue. For tens of millions of years at minimum, and possibly for hundreds of millions. The water at the bottom of the Mariana Trench is itself ancient. It has not touched sunlight in a very long time. The deep ocean is ventilated, refreshed with new water from the surface, by a process called thermohaline circulation, in which cold, dense water sinking in the polar regions flows along the bottom of the ocean toward the equator, gradually mixing with the warmer water above. But this circulation is slow, and the water at the very bottom of the deep trenches is among the last to be refreshed. Some estimates suggest that the water currently resting at the bottom of the Challenger Deep last saw the surface of the ocean somewhere in the North Atlantic, perhaps 1,000 years ago, during the Middle Ages, when European civilization was building its first universities and the Mongol Empire was expanding across Asia. The water has been traveling ever since sinking in the cold North Atlantic, flowing south along the bottom of the Atlantic circling Antarctica, entering the Indian and Pacific Oceans, and finally arriving, after a transit of thousands of kilometers and hundreds of years, at the bottom of the Mariana Trench, where it sits cold and still waiting to be displaced by the next pulse of deep water from the surface, which is itself already a century or more away. For the organisms living in this water, the passage of 1,000 years of ocean circulation is not history, it is the present. The water around them is medieval water, water that carries with it the temperature and the chemistry and the dissolved gases of an era when the surface world was unrecognizably different from what it is today. And they are living in it the way we live in air, breathing it, absorbing it, depending on it for every chemical reaction that keeps them alive, without any awareness that it came from somewhere else, or that it has been traveling toward them for 10 centuries. And still, the trench has more to show us. There is another creature in the Mariana Trench that deserves attention, not because it holds any depth records or any records for biochemical extremity, but because it is in its own quiet and faintly absurd way one of the most unusual animals in the ocean. This is the deep-sea holothurian, the sea cucumber of the Hadal zone, and its existence at the bottom of the deepest trench on Earth is a reminder that life does not always take the forms we expect, even in the most extreme environments. Sea cucumbers are echinoderms, relatives of starfish and sea urchins, and they are among the most successful deep-sea animals on the planet, found at virtually every depth from the shallow intertidal zone, where they can be picked up by hand at low tide, to the Hadal trenches, where they feed on sediment in total darkness under more than 1,000 atmospheres of pressure. There are more than 1,700 known species, and they occupy an astonishing range of marine habitats, from warm tropical reefs to the freezing waters of the Antarctic shelf, to the flat, featureless expanses of the abyssal plain, where they are often the most visible animals, plowing slowly through the mud in herds that can stretch for hundreds of meters across the seafloor. The species found in the deepest parts of the Mariana Trench are unlike their shallow water relatives in almost every visible respect. They are typically small, a few centimeters to perhaps 15 centimeters long, and they are transparent or translucent, their bodies glowing with a faint, unexplained bioluminescence, a luminous violet or pale blue light that emanates from their tissues for purposes that scientists have not yet been able to determine. Some resemble soft legless worms, moving slowly through the mud with ring-shaped mouths that ingest sediment and extract whatever organic particles and microbial biomass it contains the way an earthworm processes soil, before expelling the remainder as a trail of fecal casts on the seafloor behind them. Others are more globular, drifting just above the sediment like pale gelatinous balloons, feeding on the thin film of organic material that coats every surface at the bottom of the trench. And one group of deep-sea sea cucumbers, the genus Enypniastes, has earned the popular name headless chicken monster, a name that, while undignified, is an accurate description of the animal's appearance when it swims. Unlike most sea cucumbers, which spend their lives crawling across the seafloor at speeds that can only be described as glacial, Enypniastes can lift itself off the bottom using a large web-like membrane that extends from its body, and swim through the water column with strong rhythmic flexing motions that propel it through the darkness in a way that looks from certain angles remarkably like a plucked chicken tumbling through black water. It is not a graceful animal. It is not a dignified animal, but it is alive, and it is alive at the bottom of the deepest ocean on the planet, and that, by any measure, is an achievement worth acknowledging. The Hadal sea cucumbers feed on the sediment the way cattle graze on grass, slowly, continuously processing vast quantities of material through their digestive systems, extracting the organic carbon and the microbial biomass that the mud contains, and leaving behind trails of processed sediment that are in their own way a form of ecological service. Because the microbes that colonize the sea cucumber's fecal casts are different from the microbes in the surrounding sediment, often more abundant and more metabolically active, and other organisms, amphipods, nematodes. Foraminiferans colonize these fecal trails, feeding on the enriched microbial communities that the sea cucumber has created simply by eating and excreting. Energy moves through the trench ecosystem in a cycle that begins with the chemistry of the earth, passes through the microbes in the sediment, enters the sea cucumber's body, exits as waste supports new microbial growth, and feeds the amphipods and the snailfish above them, each organism occupying a position in a food web that exists entirely without sunlight, powered from below rather than from above. But we are still not deep. There is a dimension of the Mariana Trench that is rarely discussed, but that shapes every aspect of life within it, and that is sound. In the deep ocean, sound travels differently than it does on the surface, and in the Hadal zone, the acoustic environment is unlike anything most people have ever considered. Water is approximately 800 times denser than air, and sound travels through it roughly 4.5 times faster. Approximately 1/500 meters per second in seawater, compared to about 340 meters per second in air at sea level. This means that sounds generated anywhere in the ocean, the calls of baleen whales, the rumble of ship propellers, the crashing of waves during storms, the grinding of tectonic plates along fault lines, propagate through the water column with extraordinary efficiency, traveling vast distances with relatively little loss of energy, bouncing off the seafloor and the surface and the temperature gradients in between. And some of these sounds reach the bottom of the Mariana Trench, having traveled from the other side of the planet. In 2015, researchers from the National Oceanic and Atmospheric Administration deployed a hydrophone, a specialized underwater microphone enclosed in a titanium pressure housing, to the bottom of the Challenger Deep for a period of 23 days. The instrument recorded continuously, and what it captured surprised the scientific team. They had expected relative silence, or at most the faint rumble of geological activity. What they found was a continuous, complex soundscape. The low-frequency moans of baleen whales, sounds produced thousands of kilometers away, in the breeding and feeding grounds of the Pacific. The mechanical drone of distant ship propellers the sharp, impulsive crack of earthquakes originating from the active subduction zone beneath the trench, and the deep, almost subsonic rumble of ocean currents moving across the seafloor, carrying with them a faint but measurable vibration that persisted at all times, day and night, without interruption. The Mariana Trench, the deepest and one of the most isolated places on Earth, is not acoustically quiet. It hears everything that happens in the ocean above it, filtered through 11 kilometers of water, attenuated and transformed, but never entirely silenced. For the organisms that live at the bottom, particularly the snailfish and the amphipods, which possess organs sensitive to vibration and pressure waves, these sounds may function as a form of environmental information, a way of sensing, without sight, the state of the world around them, the presence of storms on the surface, the movements of large animals in the water column above, the geological tremors in the crust beneath them. In a world without light, sound, and vibration are the closest equivalents to sight. And the acoustic richness of the deep trench, far richer than anyone expected before the hydrophone was deployed, suggests that the sensory world of hadal organisms may be more complex, more textured, more information-rich than the darkness and the silence of the deep might lead us to believe. And now, we descend to the quietest life of all. Beneath the amphipods and the sea cucumbers and the xenophyrophores. Beneath the visible fauna of the trench, beneath even the chemosynthetic communities, on the sediment surface there is another world entirely. A world that exists inside the sediment itself, in the spaces between grains of mud and silt in the pores, and fractures of the rock below in the basalt, and serpentinized peridotite of the subducting plate. This is the deep biosphere, the subsurface realm of microorganisms, and it is here at the very bottom of the Mariana Trench and beneath it that life takes its most extreme and most ancient form. When James Cameron descended to the bottom of the Challenger Deep in March 2012, one of the primary scientific objectives of his mission was to collect sediment samples for microbiological analysis. What those samples revealed when examined in laboratories on the surface was that the sediment at the bottom of the Challenger Deep is teeming with microbial life, bacteria and archaea, the two domains of single-celled life that predate all plants, all animals, all fungi, all complex life on Earth by billions of years, present in concentrations that the examining scientists described as surprisingly high for an environment where the pressure exceeds 100 atmospheres and the temperature hovers just above freezing. These microbes were not dormant. They were metabolically active, consuming organic carbon that had settled from the water column above, and in some cases, deriving energy from chemical reactions with the minerals in the sediment itself, from the slow oxidation of iron, from the reduction of sulfate, from the metabolism of methane and hydrogen that seeped up from the deeper layers of rock beneath them. The sediment at the bottom of the Mariana Trench accumulates at a rate of approximately 1 centimeter per 1,000 years. This number deserves a moment of consideration, because it reframes everything about the environment in which these microbes live. 1 centimeter per 1,000 years means that if you pushed a probe 1 meter into the sediment at the bottom of the Challenger Deep, the material at the tip of the probe would have settled there roughly 100,000 years ago, before the last great ice age reached its maximum extent, before the first permanent human settlements were built, before agriculture, before writing, before the wheel, before any of the events that constitute what we call human history. And the microbes living at that depth, in that ancient sediment, have been there for all of that time, metabolizing so slowly that a single cell division, the most basic act of biological reproduction, might occur once every 100 years, or once every 1,000 years, or even less frequently. Deeper still, in the rock beneath the sediment in the fractured basalt and the chemically altered minerals of the oceanic crust, microorganisms have been detected at depths of more than 2 kilometers below the seafloor. In environments where the temperature exceeds 100 degrees Celsius and the available energy is measured in amounts so small that they barely register on the most sensitive instruments. These organisms are not feeding on organic material that fell from the surface. They are feeding on the planet itself, on the chemical energy released by the reaction of water with iron-bearing minerals in the rock. Reactions that produce hydrogen gas, which the microbes oxidize to fuel their metabolism, or that produce methane, which other microbes consume. They are living on geology, and they are doing it so slowly that a single generation of these organisms can outlast the entire recorded history of human civilization. This is a perspective that is worth sitting with, because it redefines what we think of as life. We think of life as something active, something that moves, something that grows and reproduces on timescales we can observe. A bacterium that divides every 20 minutes, a dandelion that grows from seed to flower in a single season, a human being whose heart beats once a second, and whose children are born after 9 months, and whose entire lifespan from first breath to last occupies less than a century. But in the deep biosphere beneath the Mariana Trench, life operates on a different clock. A geological clock, a clock that measures time not in minutes or years, but in centuries and millennia. These organisms do not rush. They do not compete in the way that surface organisms compete. They simply persist century after century, drawing energy from the slow chemistry of the rocks around them, indifferent to everything that happens above, to the ice ages that come and go, to the species that rise and fall, to the civilizations that build and crumble. None of it matters, because none of it reaches them. And still, the trench has more to tell us. There is an organism that does not live in the Mariana Trench itself, but that offers a parallel so illuminating it is worth mentioning here, because it captures something essential about the nature of deep-sea life that the trench's own inhabitants in their inaccessibility cannot easily convey. The Greenland shark, Somniosus microcephalus, is a large, slow-moving shark that inhabits the cold, deep waters of the North Atlantic and the Arctic, typically at depths of 200 to 600 meters, though individuals have been recorded at depths of more than 2,000 meters. It is not the deepest living shark, and it does not approach the pressures of the Hadal zone, but it holds a distinction that no other vertebrate on Earth can claim. It is by a significant margin the longest-lived vertebrate ever documented. Radiocarbon dating of the eye lens proteins of Greenland sharks, proteins that are formed before birth and are never replaced, has shown that these animals can live for at least 250 years, and possibly as long as 500 years, making the oldest known individuals contemporary with the Renaissance, with the age of European colonization, with the foundation of the first permanent European settlements in North America. A Greenland shark swimming in the deep Atlantic today may have been born around the year 1600 or 1550, or perhaps even earlier. Before Shakespeare wrote his first play, before Galileo looked through his first telescope, before Isaac Newton was born. These sharks have been swimming slowly through the cold, deep ocean for the entire span of modern human history, and they are still out there doing what they have always done, indifferent to everything that has happened on the surface. The Greenland shark is not a creature of the Mariana Trench, but it embodies the same principle that defines life at the bottom of the trench. The principle of patience, of persistence, of existing on timescales that dwarf anything the surface world considers normal. The organisms at the bottom of the Challenger Deep may not live for 500 years as individuals. The lifespans of hadal amphipods and snailfish are not well known, and are probably measured in years or decades rather than centuries. But as lineages, as populations, as communities, they have persisted for tens of millions of years. And the microbes beneath them have persisted for hundreds of millions, operating on a timescale that makes even the Greenland shark seem brief. There is something about these deep ocean organisms, about the shark in the cold Atlantic, and the amphipod in the Challenger Deep, and the microbe in the ancient sediment, that resists the human tendency to measure everything by the clock of a human lifetime. They are not aware of us. They are not affected by us, or they were not until very recently. They exist in a parallel time, a slower time, a time in which the events that define human history, wars, revolutions, the rise and fall of empires, are imperceptible fluctuations on the surface of a world that in its deeper layers continues unchanged. And that is the paradox of the Mariana Trench. It is the most extreme environment on the surface of the planet, and yet the life within it is among the most stable, the most persistent, the most enduring. The surface world is dynamic. Species come and go. Ecosystems collapse and rebuild, climates shift from glacial to tropical and back again. But the deep ocean, and especially the hadal trenches, change slowly or hardly at all. And the organisms that have adapted to these stable extreme conditions are not pioneers clinging to the edge of survival, but ancient residents of one of the most unchanging environments on Earth. The pressure has been the same for millions of years. The temperature has been the same. The darkness has been the same. The chemistry of the water and the sediment has changed only slightly, and the organisms that live there have had all of that time to optimize, to refine, to become perfectly, exquisitely suited to a place that never asks them to adapt to anything new. The deep ocean, in this sense, is not the frontier, it is the establishment. It is the old world, the original world, the world that was there before the surface became interesting, before the continents dried out and greened up and filled with running animals and flying birds, before the shallow seas erupted with the colorful complexity of reef life. The deep ocean was the first habitat. And the organisms that live there, the chemosynthetic bacteria, the foraminifera, the ancient lineages of crustaceans and worms, and single-celled giants, are the descendants of the first life, carrying forward a biological tradition that is older than any tradition on land. And they are carrying it forward still, right now, at the bottom of the Mariana Trench. In the same way, they have been carrying it forward for hundreds of millions of years. But here's what most people never consider about the trench. The darkness at the bottom of the Mariana Trench is not simply the absence of light. It is a physical condition with specific properties, and it has shaped every organism that lives in it in ways that go beyond the simple loss of vision. In the photic zone of the ocean, the upper 200 meters, where sunlight penetrates, light drives everything. It powers photosynthesis. It enables vision. It allows predators to spot prey and prey to spot predators. It triggers the daily vertical migration of zooplankton, one of the largest mass movements of organisms on the planet. In the twilight zone, 200 to 1,000 meters, light fades to a faint blue-green glimmer, and organisms begin to produce their own light through bioluminescence, using chemical reactions to generate photons for communication, for camouflage, for luring prey, for startling predators. In the bathypelagic zone, 1,000 to 4,000 meters, even bioluminescence becomes less common, and the darkness deepens into something that is genuinely difficult for a surface-dwelling mind to imagine. a darkness so complete, so total, so unbroken by any external source of light that the human eye, even after hours of adaptation, would see nothing at all. At the bottom of the Mariana Trench, more than 10,000 meters below the surface, the darkness has been absolute for as long as the trench has existed. No photon of sunlight has ever reached this depth. The only light that has ever occurred here is bioluminescence, the faint flickering, internally generated light of organisms that carry their own chemistry with them, and even this is rare in the Hadal zone, because bioluminescence is metabolically expensive, and energy at these depths is scarce. The organisms of the Mariana Trench have adapted to this darkness in different ways. The snailfish, as we discussed, has reduced its eyes to vestigial dots and replaced vision with a suite of mechanical and chemical senses. The lateral line system, which detects pressure waves in the water, and chemoreception, which detects dissolved molecules at concentrations so low that they are measured in parts per trillion. The amphipods retain rudimentary eyes, small, yellowish patches on their heads that may detect the faintest bioluminescent signals, but their primary sensory world is chemical and mechanical, a world of scent and vibration, rather than of light and shadow. The xenophyrophores have no sensory organs of any kind. Because a single cell does not have organs, they interact with their environment through the direct contact of their pseudopodia with the sediment, feeling their way forward grain by grain, without sight, without hearing, without any sense that we would recognize. The darkness of the Mariana Trench is not an absence. It is a presence, a defining condition that has shaped the bodies, the senses, the behaviors, and the evolutionary trajectories of every organism that lives in it. It is the single most consistent feature of the trench environment, more constant than the temperature, more uniform than the pressure, more enduring than the composition of the sediment. The darkness was there before the organisms arrived, and it will be there after they are gone. And everything that lives in the trench lives within it, shaped by it, adapted to it, unable to imagine anything else. The Mariana Trench is not one habitat. It is many. The crescent-shaped depression spans 550 kilometers of the Western Pacific, and along its length the geology, the depth, the temperature, the sediment composition, and the biological communities vary considerably from the relatively well-studied Challenger Deep at the southern end to the Serena Deep, roughly 200 kilometers to the northeast, to the unnamed and unexplored basins that lie along the northern and central sections of the trench places, where no camera, no submersible, no instrument of any kind has ever touched the bottom. Recent research has suggested that the Mariana Trench should not be thought of as a single continuous ecosystem, but as a series of at least 5 isolated habitat areas, each separated from the others by ridges, slopes, and variations in depth that function as ecological barriers, and each potentially harboring species found nowhere else on Earth. This finding, published by researchers affiliated with the Hades program, reframed the trench from a single abyss to a chain of isolated basins, each one a separate laboratory of evolution, each one running its own experiment in deep-sea adaptation. This isolation matters profoundly, because in the Hadal zone, the trenches function as ecological islands. An amphipod species adapted to life at 10,000 meters in the Challenger Deep cannot simply swim to the Serena Deep 200 kilometers away, because between the two basins lies a ridge or a slope, where the depth is shallower, perhaps only 7,000 or 8,000 meters. And for an organism adapted to 10,000 meters, that difference in pressure, though invisible to a human observer, represents a physiological barrier as real as a mountain range. To cross from one basin to another, the amphipod would have to ascend through thousands of meters of reduced pressure. A transition that its pressure-adapted cells cannot survive, and so it stays confined to its basin, reproducing in isolation, evolving independently, accumulating genetic differences from the amphipods in the next basin over. The same way that populations of finches on different islands in the Galapagos accumulated differences in beak shape and body size over thousands of generations of isolation. At a larger scale, the same principle applies between trenches. An amphipod adapted to life at 10,000 meters in the Mariana Trench cannot cross to the Tonga Trench, or the Kermadec Trench, or the Philippine Trench. Because to do so, it would have to traverse thousands of kilometers of open ocean at depths of 4,000 to 5,000 meters, the average depth of the ocean floor, where the pressure is only about half of what it has evolved to withstand, and where the temperature, the water chemistry, and the biological community are as foreign to it as the surface of a different planet. The deep-sea trenches are connected geographically. They all lie on the floor of the same ocean, but they are separated biologically by barriers of pressure that are for the organisms inside them absolutely impassable. This has profound implications for the diversity of life in the Hadal Zone. If each trench is an evolutionary island, and if each basin within a trench is a sub-island, then the Hadal Zone is not a single ecosystem, but a vast archipelago of isolated habitats, each one producing its own unique species through millions of years of independent evolution. And since the Hadal Zone has barely been explored, fewer than 30 trenches have been sampled biologically, and most of those only at a few sites. The number of species that remain undiscovered is, by any reasonable estimate, enormous. And now we come to the human story of this place, to the small handful of people who have descended through those barriers and seen the bottom of the Mariana Trench with their own eyes. The HMS Challenger expedition of 1872 to 1876 was one of the foundational voyages of modern oceanography, and it is from this expedition that the deepest point of the Mariana Trench takes its name. The Challenger was a British Royal Navy corvette originally designed for war, converted for scientific research by removing most of its guns and fitting it with laboratories, dredging equipment, and storage for thousands of biological specimens. The ship spent 3.5 years circling the globe, covering more than 127,000 kilometers, and its crew, naval officers, sailors, and a team of scientists led by the naturalist Charles Wyville Thomson, measured ocean depths and temperatures at 362 stations across the world's oceans, using weighted hemp lines lowered from the ship's deck. A slow and labor-intensive process that could take hours for a single measurement. In March 1875, while crossing the Western Pacific, near the Mariana Islands, the crew sounded a depth of 184 meters. The deepest measurement of the entire expedition, and the trench was named in honor of the ship. In 1951, a second British vessel, HMS Challenger II, returned to the region and used echo sounding, a far more precise and efficient technique that relies on timed sound pulses rather than weighted lines, to measure a depth of approximately 900 meters, confirming that the Mariana Trench contained the deepest known water on the planet. The deepest point was named Challenger Deep in honor of both ships. The first human beings to reach the bottom were Jacques Piccard, a Swiss oceanographer and engineer whose father, Auguste Piccard, had pioneered stratospheric balloon flight, and Lieutenant Don Walsh of the United States Navy. On January 23, 1960, they descended in the bathyscaphe Trieste, a vessel unlike any submarine before or since, a large gasoline-filled float for buoyancy with a small spherical pressure hull just barely large enough for 2 men attached to its underside, and iron shot stored in hoppers for ballast released electrically to allow the vessel to ascend. The Trieste was not maneuverable. It could go down and it could come up, and that was essentially all it could do. Picard and Walsh sealed themselves inside the pressure sphere at 8:23 in the morning and began their descent. The sphere was made of steel with walls roughly 12 centimeters thick, and its single window was a cone of plexiglass set into the hull, narrowing from the outside in, so that the pressure of the water would press the cone more tightly into its seat as the vessel descended, creating a seal that became stronger with depth. For nearly 5 hours they sank through the water column, watching the light fade and disappear, feeling the cold seep through the hull, hearing the metal groan and pop. As the pressure increased, the sounds amplified by the steel of the sphere transmitted directly through the hull into the small cramped space where they sat side by side, barely able to move, breathing recycled air and waiting. At approximately 9,000 meters, they heard a sharp crack, a loud, sudden noise that shook the entire vessel. One of the outer windows of the viewing port had developed a fissure. They paused the descent, examined the window as best they could from the inside, concluded that the crack was in the outer layer and that the structural integrity of the inner plexiglass cone had not been compromised, and continued downward. At a depth later corrected to approximately 10,916 meters, the Trieste touched bottom, raising a cloud of fine, light-colored sediment that obscured the window for several minutes. When the sediment settled, Picard and Walsh saw a flat, pale, featureless floor. Picard later described it as resembling a desert made of diatomaceous ooze, and remarkably, they reported seeing what appeared to be a flatfish about 30 centimeters long lying on the bottom. This observation was later questioned by scientists, who doubted that a true flatfish, a bony fish with laterally compressed body, could survive at that depth, and the identification has never been confirmed. They spent approximately 20 minutes on the bottom before releasing ballast and beginning their ascent, which took 3 hours and 15 minutes. For more than 50 years after Picard and Walsh's dive, no human being returned to the bottom of the Challenger Deep. The Trieste never dove that deep again. No other vessel was capable of replicating the feat. The deepest point on Earth became a place that humanity had visited once briefly and then abandoned, not because it was uninteresting, but because the technology to reach it reliably and safely did not exist. And the cost of developing that technology was, for most governments and research institutions, prohibitive. Then, on March 26, 2012, the filmmaker James Cameron descended alone in the Deep-Sea Challenger, a vertically oriented submersible he had spent 7 years helping to develop, reaching a depth of 10,908 meters. Cameron spent approximately 3 hours on the bottom, collecting sediment samples with a robotic arm, filming the seafloor with high-definition cameras mounted on the exterior of the vessel, and looking out through a small viewport at a landscape he later described as desolate and alien, a featureless plain of pale sediment stretching to the edge of his lights in every direction, apparently devoid of large life, though the sediment samples he collected would later reveal abundant microbial communities invisible to the naked eye. The Deep-Sea Challenger made one dive to the Challenger Deep and was subsequently retired, its pressure hull having reached the limits of its designed operational life. And then, in April and May of 2019, everything changed. Victor Vescovo, an American private equity investor, retired United States Navy Reserve officer, and extreme explorer who had already climbed the highest peak on every continent and skied to both the North and South Poles, descended to the bottom of the Challenger Deep in the DSV limiting factor, a two-person submersible built by Triton Submarines in Florida and engineered for repeated dives to any depth in any ocean on the planet. On his first dive, on April 28, 2019, Vescovo reached a depth of 10,928 meters, 16 meters, deeper than any previous human visit, spending 4 hours on the bottom. He returned the next day, and the day after that. Over the course of 10 days, Vescovo made 5 dives into the Mariana Trench, 4 into the Challenger Deep, and 1 into the Serena Deep, the latter being the first manned descent to that basin. On the 5th dive, he was accompanied by Dr. Alan Jameson, the expedition's chief scientist, and together they spent 3 hours surveying the geology and biology of the Serena Deep, collecting the deepest piece of mantle rock ever recovered from the inner slope of the trench. What made the limiting factor different from every previous deep-sea vehicle was not that it could reach the bottom. The Trieste had done that in 1960, the deep-sea Challenger in 2012, but that it could go back, and back, and back again. The submersible was built from titanium, designed to withstand more than 16,000 pounds per square inch indefinitely, and it was the first full ocean depth submersible to receive commercial certification from an independent classification society, meaning that it was tested, inspected, and certified as safe for repeated operation at any depth in the ocean. Vescovo himself has now dived the Challenger Deep more than 15 times, and the limiting factor remains, as of now, one of only 2 submersibles in the world capable of reaching the deepest point on the planet. The era of single, heroic, unrepeatable dives to the bottom of the Mariana Trench is over. The era of routine, systematic, scientific exploration of the deepest places on Earth has begun. And with it comes the possibility, the certainty really, that the discoveries made so far represent only a small fraction of what the trench contains. But we are still not finished. There is one more thing you need to understand about the life in the Mariana Trench, and it is perhaps the most important thing of all. We do not know most of it. Less than 5% of the ocean floor has been directly observed by human eyes or instruments. The Hadal Zone, the ocean below 6,000 meters, comprising all of the world's deep-sea trenches, is among the least explored environments on the planet, and the Mariana Trench, despite its fame as the deepest place on Earth, has been visited by fewer than 30 human beings in its entire history. More than 700 people have traveled to space. 12 have walked on the surface of the moon. But the number of humans who have descended to the bottom of the Challenger Deep can be counted on 2 hands, and most of the trench, the vast majority of its 550-kilometer length and its 69-kilometer width, has never been seen by any human instrument at all. The scientific community estimates that between 1 and 2 million marine species exist in the world's oceans, but only approximately 250,000 have been formally described, classified, and given names. In the Hadal Zone, specifically, new species are discovered with virtually every expedition that descends. Because the technology to reach these depths has only existed for a few decades, because the number of expeditions has been very small, and because the organisms living there have had millions of years to diversify into forms that no one on the surface has ever imagined. In 2014, the HADES program, Hadal Ecosystem Studies, conducted its first major expedition to the Mariana Trench aboard the research vessel Falkor. Led by co-chief scientists Jeff Drazen and Patty Fryer of the University of Hawaii. The expedition used 5 deep-sea lander systems deployed at carefully selected depths along the trench to sample the full range of the Hadal environment, rather than focusing exclusively on the deepest point, an approach that Drazen compared to studying a mountain by looking at all its elevations rather than just its summit. The expedition set multiple new records, including the discovery of what was then the deepest fish ever observed, a translucent snailfish filmed at 8,143 meters, and the documentation of the highest density of supergent amphipods ever recorded. But perhaps more significant than any individual record was the expedition's demonstration that every depth level in the trench, from 5,000 meters to the bottom, harbored distinct biological communities, with species compositions that changed markedly over relatively short depth intervals, suggesting that the biodiversity of the Mariana Trench is far greater than the handful of species documented to date would indicate. Every expedition that has descended into the Mariana Trench has returned with organisms that had never been seen before. New species of amphipods, new species of snailfish, new genera of xenophyrophores, new microbial lineages with no close relatives in any known genetic database. And each of these discoveries has come from a tiny sliver of the trench, a single dive site, a single lander deployment. A few hours of observation in a place that has existed for 50 million years. The gap between what we have seen and what remains to be seen is not a small gap. It is an abyss, not a metaphorical one, but a literal one, 2,550 kilometers long and nearly 11 kilometers deep, and most of it is unknown. We do not know how many species live in the Mariana Trench. We do not know the full extent of the chemosynthetic communities recently discovered by the Fendouge expeditions. We do not know what lives in the sediment below the seafloor, or in the fractured rock of the subducting plate, or in the unexplored northern and central sections of the trench where no lander has ever been placed and no camera has ever recorded an image. We know what we have found in the tiny fraction of the trench that we have been able to reach, and we know with reasonable certainty that it represents only a small sample of what exists. The rest remains unknown, and that unknowing is not a failure of science or a gap in funding or a limitation of ambition. It is a fact about the scale of the Earth and the depth of the ocean, and about the extraordinary difficulty of exploring places where the pressure destroys most instruments, the darkness defeats most cameras, and the distance from the surface makes every dive an expedition that costs hundreds of thousands of dollars and requires months of preparation. The Mariana Trench is right here on this planet, less than 11 kilometers from the surface, closer than many international flights, and we know less about it than we know about the surface of Mars. And now the trench is changing, in ways that are only beginning to be understood. During his dives in 2019, Victor Vescovo observed what many had already suspected, but few had seen with their own eyes. Human-made debris on the floor of the Challenger Deep. A plastic bag, candy wrappers, fragments of material that had no business being at the bottom of the deepest ocean on the planet, but that had arrived there nonetheless, carried down by gravity and time and currents from the surface world above. It was not the first time plastic had been documented in the Mariana Trench. In 2018, a plastic bag had been photographed at extreme depth in the trench, and subsequent studies have confirmed that microplastics, tiny fragments of degraded plastic, some smaller than a grain of sand fibers, from synthetic clothing particles from packaging, remnants of bottles and bags and films that were used once and discarded thousands of kilometers away, are present at all sample depths in the ocean, including the Hadal Zone. They have been found in the guts of amphipods collected from the deepest trench sites in the sediment at the bottom of the Challenger Deep, and in the water column at every depth between the surface and the floor. These particles arrive at the bottom the same way everything else does, slowly carried down by the same rain of material that carries marine snow to the seafloor, settling through nearly 11 kilometers of water over weeks and months until they come to rest in a place where no human hand placed them, where the organisms that encounter them have had no evolutionary exposure to synthetic polymers, and where the consequences of their presence are as yet unknown, because no one has been studying hadal ecosystems long enough to establish a baseline against which change could be measured. The trench may also be affected in the coming decades by deep-sea mining. While mining operations are more likely to target the polymetallic nodules and cobalt-rich crusts found on the abyssal plains at depths of 4,000 to 6,000 meters, rather than the hadal trenches directly. The disruption of deep-sea sediment and the discharge of sediment plumes into the water column from mining activities could alter the quality and quantity of the marine snow that falls into the trenches, affecting the food supply of the amphipods, the sea cucumbers, the xenophyrophores, and all the other organisms that depend on this material for survival. Xenophyrophores in particular, fragile, sediment-dependent, unable to move away from disturbance, are classified as indicators of vulnerable marine ecosystems by the Food and Agriculture Organization of the United Nations, a designation that reflects both their ecological importance and their susceptibility to destruction. The creatures of the Mariana Trench survived the Permian-Triassic extinction 252 million years ago, which eliminated roughly 96% of all marine species. They survived the Cretaceous-Paleogene extinction 66 million years ago, which killed the dinosaurs. They survived every ice age, every volcanic cataclysm, every shift in ocean chemistry that has occurred in the geological record. Whether they can survive the slow, accumulating pressure of modern human industrial activity, the plastics, the chemicals, the noise, the potential for mining, the acidification of the ocean as carbon dioxide concentrations rise in the atmosphere, is a question that does not yet have an answer, because the science of hadal ecology is still in its infancy. The data too sparse, and the depths too poorly explored for anyone to make predictions with confidence. And there is a geological dimension to the trench that gives context to all of this. A reminder that the Mariana Trench itself is not permanent. The Pacific Plate is still moving, still sinking beneath the Mariana Plate at a rate of roughly 6 to 10 centimeters per year, and the material that makes up the trench floor, the sediment, the mud, the rock, and everything living in and on it, is being carried slowly, imperceptibly, toward the interior of the planet. The organisms at the bottom of the Mariana Trench are living on a conveyor belt, a surface that is moving, century by century, toward the heat and the pressure of the upper mantle, where the rock will be melted and recycled and eventually return to the surface as new crust at a mid-ocean ridge somewhere. Millions of years from now, in a cycle that has been repeating since the Earth's interior first began to convect roughly 4 billion years ago, the trench will continue to exist for as long as subduction continues at this boundary, which will be, by geological estimates, tens of millions of years at minimum. But the specific patch of seafloor where the amphipods are swarming tonight, the specific sediment where the xenophyrophores are building their tests, the specific rock where the microbes are metabolizing in their geological patience, all of it is moving, all of it is temporary, all of it will eventually be consumed by the planet's interior and replaced by new material. New sediment, new organisms that will colonize the new surface and begin the cycle again. And now we come to the end of tonight, to the place where what we have learned settles into something you can carry with you as you rest. Remember that the Mariana Trench is not empty. that it is home to amphipods that swarm by the thousands in total darkness, feeding on the rain of particles from above. Their cells packed with molecules that evolved specifically to function under more than 1,000 atmospheres of pressure molecules that would not work on the surface, that are designed for the deep and for the deep alone. Remember that single-celled organisms the size of a human fist sit on the sediment of the trench floor, building elaborate shells from the debris around them, from grains of sand from the broken tests of other organisms, from bits of volcanic glass and sponge spicules and mineral fragments, hosting entire communities of smaller creatures that depend on them for habitat, in a world where solid surfaces barely exist. Remember that the deepest fish on Earth, the snailfish pale and translucent and nearly blind, has been adapting to the conditions of the Hadal zone. For 20 million years, its skull left unfused, its scales abandoned. Its eyes reduced to dots, every aspect of its body shaped by an environment that no surface organism could endure, and that it feeds on the amphipods below it with a hunting sense built not on sight, but on vibration and chemistry, and the delicate perception of pressure waves in the water. Remember that, beneath the visible life, there are organisms that derive their energy not from the sun, but from the chemistry of the planet itself, from methane rising through fractures in the seafloor, from hydrogen released by the reaction of water with rock deep in the subducting plate, from sulfur compounds seeping from the interior of the Earth, and that these organisms have been practicing this form of life for hundreds of millions of years, possibly, for as long as life itself has existed. In a biosphere that operates on principles so different from the surface world that discovering it was like discovering a second way of being alive on the same planet. And remember the microbes, the slowest, the smallest, the most patient organisms of all, living in the sediment and the rock beneath the trench, dividing once a century or once a millennium, feeding on the chemistry of the Earth's interior, with a persistence so profound that no human word adequately describes it. In a world where the passage of 10,000 years leaves no more trace than the passage of 10 seconds leaves in hours. And it is still there. Right now, as you breathe, as you rest, as you drift towards sleep, the Mariana Trench is doing what it has always done. The amphipods are swarming over the sediment in total darkness, their translucent bodies flicking through cold water, sensing food with chemical receptors that can detect a particle of marine snow from meters away. The xenophophores are slowly extending their pseudopodia into the mud, gathering another grain of sand for the test, adding another fragment to a structure that has been growing for years or decades, one grain at a time, in the same way that the sediment itself has been accumulating. 1 centimeter per 1,000 years since before the last ice age. The snailfish are gliding silently through the cold water at 8,000 meters, their wing-like fins barely moving, hunting by vibration and chemical trace in a world that has never known a single photon of sunlight. The tube worms are waving their red plumes in the darkness at 9,000 meters, absorbing the methane that seeps up through the sediment, feeding the billions of bacteria inside them that convert the chemistry of the Earth into the chemistry of life. And deep in the sediment, in the fractures of the rock, in the spaces between grains of mud that settled 100,000 years ago, the microbes are still there, still metabolizing, still alive, drawing energy from the interior of the planet with a patience that makes everything on the surface seem hurried and brief and impermanent. The Mariana Trench is the deepest place on the surface of this planet. And it is full of life. Life that does not need the sun life, that does not need warmth life, that does not need anything that we as surface organisms consider essential. Life that has persisted through every catastrophe the Earth has ever produced, and will almost certainly persist through whatever comes next. Because the chemistry that sustains it is not dependent on the climate, not dependent on the atmosphere, not dependent on anything that happens on the continents or in the sky, but only on the heat of the Earth's interior, which will last for at least another billion years. The ocean at those depths is quiet and dark and still, and it does not make exceptions. Tonight we traveled from the surface to the deepest point on the planet, and we found that even there, especially there, the Earth is alive in ways that most people never imagine, and that science is only beginning to understand. Good night, and may you never think of the ocean floor as empty again.